So now let's talk about prokaryotic transcription. Nothing fancy here. We've already learned about eukaryotic transcription. Eukaryotic transcription. Where does that take place? It occurs in the nucleus. We have RNA processing. We have the addition of a poly A tail. We also have an addition of the 5' GTP cap. And now let's talk about prokaryotic transcription. Prokaryotic transcription occurs in the cytoplasm. Remember, prokaryotic do not have any membrane-bound organelles. They do not have a nucleus. They do not undergo RNA processing. And also, they do not have a poly A tail and a 5' GTP cap. Remember, these are very essential, and there's a minor difference between prokaryotic and eukaryotic. Also, one last thing, prokaryotics do transcription and translation simultaneously. Well, I spelled wrong. But anyways, transcription and translation are simultaneously done for prokaryotic cells, and eukaryotic cells do transcription first. It's exported out of the nucleus, and then we do translation. So these are the differences, and now let's start to talk about translation. But before we talk about translation, let us make up a mRNA transcript so we can understand. Okay, so 100 times drawing an mRNA transcript. 5', 3', poly A tail, 5' GTP cap, we'll just pretend. Okay, great. So, let's see. Let's make this C A G G C A U G C U A U U. Actually, I should make this longer. C U G C C C C A U and U A. Okay, so here's my messenger RNA, mature messenger RNA. It has this poly A tail, 5' GTP cap. It has this exonic sequence inside. It's ready to be exported out of the nucleus. Minor things we should remember: each 3 bases are referred to as codons or a triplet, and each codon codes for an amino acid AA, short for amino acid. I'm just going to write it down: amino acid. And how many amino acids are there in your body? There are 20 types of amino acids in your body. Please, it's important to remember that. 20 types of amino acids in the body. You do not have to memorize all these amino acids. You don't have to memorize the names of it. Over here to my left, we have a codon chart, which tells you what these codons code for, what amino acid it codes for. So I have these different codons here, and you'll notice I have all 20 amino acids here. You don't have to memorize any of them. We have phenylalanine, leucine, proline, histidine, and so on, isoleucine, and all these 20 amino acids. You do not have to memorize them. You simply have to understand how to read this codon chart, and it's very simple actually. So let's try an example. Let's just take the first codon, CAG. I don't know what that codes for. CAG, the ribosome will ultimately make what amino acid from it? Well, how do you read this chart? You start from this side over here, and then this is going to be the first base, this will be your second base, and then you locate your third base. So let's look at CAG. I have C here, I have A here, so it's going to be somewhere over here, and then I find the third amino acid, which is, third base, which is G, CAG, TL. So, on your test, if this was a Regents exam and they said, okay, here's my transcript, now please list me the amino acids. So you would write GLN, dash, let's do GCA, GCA, ALA, and then we have AUG, AUG, codes for MET, and so on and so on and so on. So it's simple. But now let's get a bit more complex and see how this actually works. So understand how to read the codon chart. Now let's take a look at this. This guy is already transported out now. Pretend he's out of the nucleus, is in the cytoplasm, and now he finds a ribosome, or she finds a ribosome. I don't know why I said he, but he finds a ribosome, and now I have a ribosome is made up of 2 subunits. Let's see how I can. I'm not going to do this justice, but. We don't have to even make it that big. Let me make this smaller. You look something like this. Small subunit. So the small subunit and the messenger RNA will first bind. The mRNA will locate the small subunit, and then the large subunit will ultimately come in. And now we have things going on that will happen. And you'll notice that, CAG goes for GLN, and GCA, what did it code for? ALA. You'll notice these different amino acids, but the ribosomes will start to translate the protein when it sees a start codon. And you should memorize, you have to memorize this. The start codon is AUG, and it's the only start codon. AUG, which codes for methionine, AUG is the start codon. Remember that. AUG is the start codon, so when the ribosomes recognize the start codon, it's going to say, okay, we're starting to now translate to protein, so now methionine, we're going to put the amino acid there, and then the following amino acids, and such and such and such, and finally, of course, hopefully we're going to code for something that says stop, and of course, we have a few codes that code for stop over here. These all code for stop. So let's see what happens in between this entire process. So an important part of the ribosome is that we have 3 different sites. We have the E site for exit, we have the P site for peptidyl, and the A for amino acyl. The E site, P site, the A site, these are not actually sites on the ribosomes. These lines are more imaginary, if you will, but this helps us understand how translation works. So once again, it's going to start from the AUG site to AUG, but how does the ribosome recognize these codons? This is where we introduce another type of RNA molecule called tRNAs. These are called transfer RNA molecules, tRNA. These are transfer RNA molecules which contain the anti-codon of the respective codon. What's a codon? Well, these are codons, codon, codon, codon. And anti-codon would be the complementary bases of the codon. So if that's AUG, then the anti-codon would be what? U, A, C, right? A pairs with U, U pairs with A, G pairs with C. This will be my anti-codon, and I will just draw a tRNA molecule that looks something like this. Looks very ugly, but whatever. And over here we have our amino acid, methionine. It's bound to the tRNA molecule, and this guy over here is going to come here, and it's going to recognize this codon. The anti-codon and codon will recognize each other. And then this methionine right here is going to get transferred onto the P site. Pay careful attention because now things get really weird and messy. Once again, so this starting codon, right, AUG, methionine, which goes from methionine, will come, this tRNA will come in, recognize this, the anti-codon with the codon, and say, okay, great, let me transfer this methionine into the P site. The tRNA comes to the P site, and now there's this very special enzyme. It's called amino acyl tRNA synthetase. Amino acyl tRNA synthetase is going to release this amino acid that's bound to the tRNA and transfer it onto the P site. So let's draw that. Actually, I drew methionine in red, but let's pretend it's blue. So here's my methionine. It's in the P site. Amino acid tRNA synthetase an enzyme has released this methionine into the P site. And now what? Well, we have many tRNA molecules floating around in the cytoplasm, and each of them contain a different anticodon. So eventually, the respective tRNA molecule that has the anticodon to this codon is going to come in. So let's draw that. So 1, 2, 3. Let's see. CUA. CUA codes for leucine. CUA codes for leucine. We're going to label this leucine. LEU. And what's the anticodon in this case? It's going to be G. We wanted A. U, so it's GAU. And this tRNA is going to come over here, recognize the codon, and then amino acid tRNA synthetase is going to place the amino acid where? Not the P site. It's going to place it on the A site. Methionine is the only amino acid that is always placed the first time in the P site. Everything else following is placed on the A site. Keep that in mind. See, this is where it gets a little bit confusing. So once again, methionine is placed in the P site. The next tRNA molecule that follows, whether it's leucine, isoleucine, proline, whatever it may be, will follow in the A site. Amino acid tRNA synthetase will release it, and these tRNA molecules will enter out of the E site. The exiting phase, tRNA molecules leave. I have an amino acid here, methionine. I have an amino acid here, leucine. Now what happens? Well, there's an enzyme called Peptidyl Transferase. So this is another enzyme you have to know. Peptidyl Transferase. Peptidyl Transferase, which is going to transfer all of the amino acids from the P site to the A site. And it's going to create a peptide bond between them. Remember, these are amino acids. What kind of bonds are between amino acids? Peptide bonds. This is a little bit of biochemistry, if you remember. Amino acids have a peptide bond between them. So, peptidyl transferase will transfer this amino acid to the A site. And now, what do we have? We have methionine and leucine with a peptide bond. And I'm going to erase this. And then what happens? The ribosome is going to shift one unit, or I should say, it's going to shift from codon to codon. And it's going to start reading and reading and reading. But what happens? Okay, so I told you these lines are imaginary. So technically, when this ribosome shifts to the right one unit, what's going to happen? Well, everything in the A site is now technically in the P site. So let's pretend this thing moved. So I'm not going to redraw this entire diagram because then it would take me forever. But okay, so this is now in the P site. We've done this. Now we need AUU. So AUU. Okay, great. It's isoleucine. Okay, so let's see. Another tRNA molecule comes in. Now let's see what happens. What's the anticodon? UAA. Please remember, when you're reading codon chart, you're reading these to find the amino acids. Don't make the common error and read the anticodon. Say, oh, hey, look, let me find the anticodon. Hey, UAA. Where's UAA? Right? That codes for stop, by the way. If that codes for stop, then we are already going to... we have 2 amino acids. This is not even a functional protein. This is no protein at all. And proteins have to be at least a minimum specific amount of number of amino acids. But this seems obviously ridiculous. Actually, this example was also a bit wrong because it's very short, but that's not the point. The point is for you to understand the process of translation and how it works. Please look at this. This is the Mrna transcript. This is what you're looking at for the codon chart. AU codes for isoleucine, ILE. What happens? Once again, nice. It's going to do what? Amino acid tRNA synthetase is going to release the amino acid bound to it to the A site once again. And then what's going to happen? peptidyl transferase is going to transfer these amino acids to this site and is going to create a peptide bond. So now we have something that looks like this, and the tRNA molecule, the empty tRNA molecule, will leave the active site. So now we have this, and then the ribosome will shift 1 unit to the right. And this process keeps happening and happening. Hopefully you get the picture now of how this process works. And so at the end you have a bunch, you have really an amino acid chain, protruding out from the ribosome. Looks pretty nice. You have a primary structure of protein, which is just a chain of amino acids. And finally, when you reach a stop codon, so in this case, well, let's just pretend, well, let's take UAA. UAA, I've reached a stop codon. Now notice, well, take important note of this. The stop codon does not code for an amino acid. It codes for a protein. It codes for something called a release factor or a releasing factor. Stop codons are not an amino acid. So stop codon codes for a releasing factor. And this releasing factor will basically hydrolyze this amino acid chain. Hydrolyze meaning it's going to add water, so it's going to break apart that bond. So it's going to hydrolyze this reaction, and now this chain of amino acids is no longer bound to the ribosome. So what does that mean? Now I have a free-floating amino acid chain in the cytoplasm. This free-floating amino acid chain in my cytoplasm is noted as my primary structure of protein. So technically, this process of translation, we haven't made protein yet, but we've entered the primary stage of protein. And if you remember in the earlier videos of biochemistry, we talk about primary structure of protein, secondary structure of protein, tertiary and quaternary structure of protein. This is how translation essentially works. Now I want to clarify something. I want to introduce a few ideas. Hopefully you understand this entire notion of translation and you understand the basics. This is what you should know. You should know peptidyl transferase. You should understand amino acid tRNA synthetase. You should understand these 3 sites. But beyond the fact of that, here's something that I would like to clarify a bit. It's not as if that there's only one AUG in each mRNA transcript. In reality, there could be an AUG here, an AUG over here, an AUG over here, and you might say, hey, how does the ribosome know when to recognize these start sites? You just told me that I have multiple start sites. Well, if I have an AUG right here, and an AUG right here, is this enough of a length of me to make a functional protein? No. So the ribosome kind of just goes ahead and jumps and gets a feel for which AUG is the best to make a functional protein. So that's one of the, that's something you should have in the back of your mind. And also this ribosome follows more of a hit and miss model. It's not perfect. Nothing is perfect. I've taught you replication of DNA, transcription of DNA. A lot of the things I've taught you is oversimplified. Translation is also definitely oversimplified, but you at least have the fundamental concept. You understand the basics now of translation. Please memorize this. Please understand how to read this chart. Do not memorize this chart by any means. You will never have to memorize this chart unless you take a biochemistry class. So that's it for translation, and we're going to sum up everything else about molecular biology in the next video.